When we are born, we're born sterile. So there are no microbes in and on our bodies when we're in our mom's womb. And as soon as we're born, we are instantaneously inoculated with trillions of microbes.
Welcome, everybody. This is Buck Joffrey with Longevity Roadmap. And today we're going to talk about about the microbiome a little bit more. This is an area that I still find very confusing. I know a lot of influencers out there that you hear about have an answer to all of what ails us through the microbiome, which obviously is not the case. It's not necessarily the case that the microbiome is not responsible for a lot of things. It absolutely is. But controlling that microbiome is another thing entirely. I don't think there's one panacea for everybody, not one probiotic, no specific panacea that's going to correct all that ails us. And so today I've got a guy who specializes in this stuff. They are doing a lot of research on, trying to figure out how the microbiome evolves with age, what goes wrong in some people and how you can potentially redirect it with various therapies. And it's not as simple, again, as simply just, grabbing a probiotic off the shelf and taking it. It's actually pretty complicated stuff, as I find this entire topic. But it's incredibly important because the microbiome is responsible for a lot of our health and therefore responsible for a lot of things that ail us as well, like inflammation in our bodies, people with inflammatory bowel disease, things like that. There's even been reports on autism that have shown benefits with fecal transplant. Anyway, really fascinating interview with Sean Gibbons, and we will have that for you right after these messages. Hey, longevity enthusiasts, it's time to take it to the next level. I've been fine-tuning my longevity regimen for years, and I look better and feel better than I did a decade ago. In fact, my blood work is even better than it was back then. And it's all because of my data-driven regimen, and it's inspired me to create a course and community just for you. It's called the Longevity Roadmap, and I urge you to check it out. If you're tired of your belly fat, tired of being tired, or just want to optimize yourself for the next 50 years, visit longevityroadmap.com. That's longevityroadmap.com. Welcome back to Longevity Roadmap. Today, my guest is Dr. Sean Gibbons, Associate Professor at the Institute for Systems Biology in Seattle and Washington Research Foundation Distinguished Investigator. He's also an affiliate faculty member at the University of Washington in bioengineering, genome sciences, and data science. He earned his PhD in biophysical sciences at the University of Chicago, completed a Fulbright in microbiology and synthetic biology at Uppsala University, and did his postdoctoral work at MIT studying ecoevolutionary dynamics of the human gut microbiome. His lab focuses on understanding how our gut microbes evolve over a lifetime and how we can design personalized interventions to promote health and longevity. Sean, welcome to the program.
Thanks, Buck, for having me.
I just want to kind of start out setting the stage here. I think a lot of your research revolves on the idea that the gut microbiome isn't just an active passenger. It is, a passive passenger. It's active. Changes with their environment, diet, etc. I guess the question for you is, I think for perspective, I was thinking about this myself. A lot of people end up with messed up microbiomes. I'm probably one of them. How are we born? Are people born with a good microbiome inevitably that can be altered and go wrong? Or what happens at birth and as we grow?
That's a great question. So I don't know how far back to go. So
I guess birth.
Yeah, birth, but even before that, right? Evolution. Yeah. We're multicellular organisms, and we're living in a microbial world, right? For about 2 billion years before eukaryotes and multicellular organisms arose, there were microbes, and they can kind of do anything, metabolically speaking. And so they colonized us just like they do everything else. And we had to learn how to live with them. We were either walking happy meals to be eaten by them, or we could cooperate with them. There's all these ways we can interact with our microbes. And so over long time periods, we've sort of evolved a stable interaction. The vast majority of these microbes in and on the human body are either benign to us or even beneficial, whereas a very, very small minority of things are pathogens. When we are born, we're born sterile. So there are no microbes in and on our bodies when we're in our mom's womb. And as soon as we're born, we are instantaneously inoculated with trillions of microbes. Mom is sort of the sourdough starter culture. She kind of gives us our first dose. We get if we're vaginally born, we're exposed to vaginal microbiota. It's also a little bit of poop there, right? And moms poop a little bit when they give birth, usually. So you really get a kind of diverse inoculum from mom. C-section born babies, their microbiomes actually look more like the skin for the first few weeks of life. So you definitely get more of an inoculation from skin associated microbes. But, as we grow and develop from infanthood to early childhood, we create sort of a self-assembled ecosystem. We're sampling microbes from our environments, from the people around us, but also from the dirt or from touching the dog's nose or what have you. And if you look at, two people's microbiota, they're very, very different. They're one of these things that makes us very unique in addition to our genome. And even identical twins who share the same genome have microbiomes that are almost as divergent as two complete strangers.
It's interesting because you think that, most children start out with, this is just my perspective, most children don't start out with issues with their gut, right? Maybe that's true, maybe it's not, but that's just my impression just in terms of how things seem to evolve. Older people seem to have more issues to develop over time. Is that true or is that just not true?
It depends, right? Like colic
Yeah.
is a common condition for babies. A lot of time that's due to bloating and gas and pain. It's hard to pin it down, but I think some amount of that is gut related. You do see that babies born via cesarean section or vaginal birth show variable outcomes. Also, kids that have different exposures to antibiotics throughout their childhood, like kids with chronic ear infections, for example, receive lots of antibiotics throughout their childhood. And that perturbs your gut microbiota in addition to everything else. We see that kids who are born via cesarean, who don't breastfeed or who are exposed to a lot of antibiotics, and all of those things can be kind of a perturbation on that early life microbiota. A lot of those kids show elevated risk for certain overactive immune system disorders like autoimmunity, asthma, allergies, food allergies, and so forth.
Yeah, the other thing that comes to mind, and I doubt there's a study on this, but you would think that you would be able to link some things in these children from their mother's microbiota. If that's if they're coming through natural childbirth and that's their first exposure, do we have any data on that?
We do. There's a lot of cool studies on this, sequencing the mom's stool and vagina and skin, and looking at the babies, and you for sure see exact strain transfers. So the exact same strain of E. coli or whatever bug that was in mom is making it into the baby, but it's less than you would expect in terms of like the total composition of the microbiome. Maybe only like a couple of percent or a few percent of the strains in the infant are coming from mom, and 90 some percent are being derived from somewhere else, and we don't quite know exactly how that sourcing is happening. But we see siblings, mom and dad, these close social interactions do give rise to exact strain sharing, whereas it's very, very unlikely that two strangers who've never physically interacted share the exact same strain.
Interesting. What are some of the most important changes to the gut microbiome as people age? And maybe that's not the right question from what you're saying, but is there a natural change or progression of the microbiome that is attributable to the aging process?
Yes. So there's a few phases that you can separate your life into. So the first is going back to being a baby. Our diet is very unique. As mammals, during that early phase, we're eating milk. That's the predominant source of energy. And in the human gut, there are these organisms called actinobacteria, and specifically bifidobacteria, which have evolved to consume the polysaccharides, the human milk oligosaccharides that are present in milk. It turns out that mom actually produces a bunch of these HMOs, or human milk oligosaccharides. A large fraction of the total energy content of milk is made up of these oligosaccharides, which the baby cannot digest. So these are molecules that we've evolved to put into milk that exclusively cultivate specific types of bacteria in the gut, in particular these bifidobacteria. And so having high levels of bifidobacteria in the first several months of life while you're breastfeeding is very healthful and very good. It suppresses pathogens. And as we transition towards solid food, you see a flip in the ecosystem where the actinobacteria decline precipitously. You get an enrichment in these other two major clades of anaerobic bacteria that belong to, I think now it's called the Bacillota phylum and the Bacteroidota phylum. Gram-positive and gram-negative anaerobes that tend to be dominant in the adult human gut. These rise into prominence by about a year or two of age, and then your microbiome kind of stabilizes into an adult composition. And that adult composition is very stable. Some people call it our second genome. Like I just sequenced my microbiome again after 7 years, and it's about identical to what it was 7 years ago. So the ecological composition of that adult microbiome is very, very stable and steady over long periods of time. And so that's most of your life. But then towards the end of your life, as you get into later middle age, like 50s, 60s, you do start to see that there is a bit of a shift that occurs. And this is actually recent work from our lab where we've characterized that there are actually kind of multiple trajectories of aging towards the end of life. And those have variable associations with health.
And I guess the question is, how much control of that do we have? I mean, that obviously leads into, a number of health related issues. How controllable are those changes?
That's a great question. And we don't quite understand the mechanistic drivers for what is driving these different signatures. So maybe I'll first describe what those signatures are.
Mm-hmm.
So when we look at data from older humans, we see there's a certain subset of people who kind of maintain a microbiome that looks like it did when they were in their 20s and 30s and 40s. You see a very high prevalence of these gram-negative anaerobes like the Bacteroidota, and then also a mixture of these Bacillota, these gram-positives. And if you maintain that composition into later life, that maintenance of a more youthful microbiome is actually associated with worse health outcomes, right? Those people aren't doing as well. Alternatively, there is this pattern that's been observed in centenarians and in healthier older people where you get this steady decline in the Bacteroidota phylum. The gram-negative anaerobes are kind of going down in their dominance in the ecosystem, and you see a relative rise in the dominance of these Bacillota, the gram-positive, like Clostridia, butyrate-producing taxa. They're kind of rising in prevalence with older and older age. What is driving this is still kind of unknown, but there are a few things happening as we age. One, we have kind of adrenopause, menopause, hormonal changes in the body. Some of these are affecting things like transit time, like how fast we're pooping. Sometimes that kind of slows down a little more as we get older. Appetite declines a little bit. So we're maybe taking in a little bit less food than we used to, and that's going to affect the microbiota. Our gut produces mucus, which lines the interior of the gut and kind of acts as a barrier or a wall between us and our microbes. Our ability to produce mucus actually declines as we get older, and so that wall starts to get thinner. The gram-negative anaerobes that I was talking about, like the Bacteroides genus, for example, many of these bugs can eat the mucus layer, And so their presence could maybe even lead to thinning of this eroding mucus layer as we age. So having lower levels of these guys could be beneficial in that context. I suspect that these variable patterns are being driven by healthy lifestyle. So people who are maintaining a high level of dietary polysaccharides are probably enriching for these Clostridial taxa with age, as the niche for the gram negatives is maybe declining due to these physiological processes that are associated with aging.
Talk about these changes that are occurring with age. Makes me wonder a little bit, again, just on this idea of like how much can you actually change the microbiome with things like, probiotics, prebiotics, postbiotics, things like that. I mean, is there a certain level of resistance to those changes that develops over time as you have that steady state microbiome? And if you try to alter it, because some people, may not have a healthy microbiome, how difficult is it actually to meaningfully change that ecosystem?
It's difficult. It's actually frustratingly difficult.
Mm.
You can definitely change your microbiome with radical shifts to your diet and lifestyle. So if you were eating a pure carnivore diet, then you switch to a high fiber vegan diet, you would definitely start to see shifts happening in the ecosystem. But there's a lot of kind of priority effects in the gut. So once an organism is in there and establishes, it becomes difficult for another competitor to dislodge it. So
Yeah.
that adds sort of a inertia or a hysteresis to the composition of the microbiome. And then, for a new bug to come in and get access to an emerging niche, it needs to go through the bottleneck of the stomach, right? So there's this bottleneck that's trying to kill everything that's coming in through our diet. So if only a few cells can make it through, they happen to hit the right spot and bloom and get in there. So you sort of have to wait around if you change your diet for these organisms to kind of get in and colonize those niches. So there's a lot of slowness to the process. And there's also uncertainty as to like, how do we rationally engineer or select prebiotics and probiotics? That's a
Yeah.
lot of actually what our lab is working on currently, are modeling tools to kind of build a digital twin or representation of the gut microbiota metabolism and ask, of all these probiotic organisms that I could add to the system, which one would I predict would be most likely to grow in your particular microbiome, in your particular dietary context? And we have some recent papers kind of showing some efficacy for those types of models. So I think that's the future, where we will have to maybe design targeted or precision prebiotic and probiotic and dietary interventions to be able to change and adapt the microbiota at will.
Well, let's jump into that a little bit more in terms of what you guys are doing. You've developed methods to essentially reconstruct dietary patterns from stool DNA, right? How do you do that? And I guess what kind of output do you get from those studies?
Yeah, so that is actually a recent method that just came out on how to infer diet from gut sequencing data. So the basic idea is, we're eating things with DNA in them. When I eat a corn on the cob, that corn has DNA, corn DNA in it. And some amount of that DNA survives passage through the digestive tract and ends up in stool. Now, the problem with that, though, is that the vast majority of DNA in stool is coming from bacteria. By weight, stool is about half bacterial cells. And by DNA content, it's something like 99.9% of all the DNA is coming from bacteria. So when you're sequencing it, it's like finding a needle on the haystack to find the corn DNA in this vast soup of microbial DNA. But we figured out a way to do it. Research scientists in the group, Christian Diener, built a bioinformatic workflow that takes in this shotgun sequencing data from DNA extracted from stool, maps it to a large database that includes something like 400 genomes of all of the plants and animals and fungi that are in the human diet that have been sequenced, that are available. We then sort of extract that information and also kind of the abundance information of how many sequences are mapping to each organism, and then reconstruct a matrix of, here are all the different organisms we're detecting in your stool, and the counts of how many DNA sequences we found from that organism as kind of a relative abundance metric for those things. And that can be used to map to a different database that contains information about, for every gram of beef, there are X number of grams of alanine and cysteine and thiamine and all these other things. So you can get kind of nutritional information from the biomass information. And together you can get both composition of diet in terms of what types of organisms you're eating, and nutritional content of diet in terms of what are all the macro and micronutrients. Then we did some benchmarking with some controlled feeding studies, and it works pretty good for certain contexts. For some things, it doesn't work so well, but we're kind of continuing to develop the method to make it better.
It sounds like, this is kind of headed towards this, I guess it's a popular term, but precision nutrition type of thing, right? Um, what does it look like? Like when you use this data, what is the ultimate goal? I mean, it looks like you're basically able to look back and see what people are eating from the microbiota. What do you do with that? Or what is the goal ultimately to apply this in sort of a precision health model?
Yes, great question. So this actually relates to a different method we've developed called MyCom, which is a portmanteau of microbial and community. It's MyCom. And it's essentially, we're taking a method called flux balance analysis, and it's a mathematical framework for taking a biochemical reaction network, like all the enzymes for all the metabolic reactions that are happening in one organism's genome. You can pull out those enzymes and build a reaction sort of matrix, and then include sort of a dietary or environmental constraint matrix. So like how much of all these nutrients are available to this biochemical network. It's like you're pouring these things into the network and looking how they filter through it and predicting how they're flowing out the other end. So we take that kind of a modeling framework and we extend it to complex multi-species communities like a gut microbiome. So we can build these models that have hundreds of organisms in them, each in their own little compartments with internal metabolic reactions occurring, transport reactions happening across their membranes, and then an external dietary milieu where they can exchange metabolites, consume and produce metabolites, and cross-feed with each other, and then feed things to the host, which is us. So it's these big unwieldy models that we can now create, and I can initialize them with sequencing data, say from your gut. So I sequence your microbiome, I see which taxa are there and their relative abundances. I can use that to initialize one of these models of your particular system. And then for the dietary input, which is another constraint we need to layer into the model, we can use what I just talked about, which another method we call metagenomic estimation of dietary intake. We can take that dietary intake estimate and we can turn it into a model accessible diet. And together we can then predict, if you eat this diet that you're currently eating, what microbial metabolites are being produced. And we have a few that we've focused on for validation. So butyrate was one we recently published on. To a first approximation, the microbiome is this bioreactor that takes dietary fiber and ferments that fiber into organic acids, right? The kind of things you taste when you drink like kombucha or eat sauerkraut, these sour tasting molecules. Our microbes are producing these things in our guts all the time. And these have a really positive, healthful effect for our bodies. Now, in the developed world, we are very much underexposed to these organic acids because we don't eat as much dietary fiber as we used to back in the old days. We probably ate 100 grams of fiber a day as hunter-gatherers, and today we're lucky to get 10 grams of fiber in the modern American diet. And so, turning up these organic acids is really key to preventing a lot of chronic diseases that seem to arise from this missing fiber in the diet, like diabetes or autoinflammation and so on. And so what we wanted to do with our model is to predict butyrate production in particular, which is one of these organic acids that have a really potent health effect in the body. The problem is, if I feed a banana to 10 people, up to 10 times different amounts of butyrate will come out at the other end. So what we did is we showed that our modeling infrastructure could predict this variability. So I can predict your butyrate and my butyrate production We validated these predictions using, we call them poop soup experiments. So we could take fresh stool from a person in the lab, incubate it in the lab with, say, adding a dietary fiber, and then measure the butyrate production, and do that for 100 different people, and then model those same interventions and show that the two align very well. So basically, to your original question, how do we use this information? If I could turn up your butyrate level at will, I could probably reduce your LDL cholesterol. I could improve your insulin sensitivity. I would lower your risk for cancer in the long run, and any number of healthful effects, right? It's this very potent molecule that has myriad effects across the body. And so at this point, we've done some of the preclinical validation. We're now thinking about moving into human clinical trials where we try to predict personalized prebiotic, probiotic, and dietary interventions that perform better than a standard of care intervention, like maybe eating a Mediterranean diet or something.
Yeah. So in this situation, is the primary, it sounds like the primary endpoint, at least for the stuff you've talked about, is butyrate production. Is that right?
If I had to pick one molecule that was maybe the most important thing that our microbiome is producing day to day, it would be butyrate. So that's why we focused on that one. And then we generated some validation data showing that we could indeed accurately predict personalized production rates for this molecule. And the downstream effect of that molecule, we were able to assess in a lot of different ways. Like there's a lot of literature on butyrate and what it does in the body, but we had a cohort where we had paired poop samples and clinical chemistries you can get measured at the doc, something like 130 of them. And we associated all of these with our predicted butyrate fluxes and found that about, 30 or 40 of these chemistries were significantly associated with predicted butyrate. And they all showed the directionality we would expect. Like if you're predicted to produce more, you have lower LDL, you have better insulin sensitivity, you have lower inflammation, so on and so forth. So, to like a clinical endpoint, what would you look at in a trial? I think you could kind of pick your poison to some extent, but I would focus on like a cardiometabolic endpoint, probably like HbA1c or blood pressure, something like that. But whatever we do, we're trying to find that first proof of concept to show that a microbiome guided personalized intervention is optimal and does better than some standard intervention.
Just specifically to butyrate, and notice it's often in various nutraceuticals now. Is there a problem with getting that butyrate to where it needs to go when you take it by mouth?
Yeah, that's like a postbiotic, they call this, right?
Yeah,
So you take a product that the microbes are making and you just manufacture that as a drug, and you just give it to people without worrying about the microbes. This is something that can be done. Actually, last week, there's some people that I know have published a paper on butyrate enemas showing that they dropped blood pressure by 10 points in a randomized placebo-controlled trial. So we know it works, right? You give a high dose enema, that works. There are studies that have shown taking pills with butyrate in them may or may not have effects. So it seems that if the butyrate is delivered to the small intestine, it doesn't necessarily have as potent of an effect as if that butyrate gets delivered in the colon, where a lot of the receptors are there for detecting that butyrate and for responding to it. So I think spatially where you deliver that butyrate is really important. So that's one challenge. The challenge for the enemas is that it stinks to high heaven, right? Butyrate is very stinky. Patients actually don't like getting butyrate enemas because they smell like a garbage can for a day after they get it. So that's a problem. And finally, I'll say that, in order to have, an optimal effect, I think you need large amounts to be produced or to be delivered continually. And short of some like tube that's like feeding it in all the time, I would argue that why not leverage our endogenous microbiota to be constantly producing it in situ?
Sure. We've been talking about, I think you mentioned this with digital twins, the concept of digital twins. Basically, there's virtual models that can test interventions before trying them in real life, right? Isn't that sort of the concept there?
Yes.
So it sounds like to me, from what you're saying so far, that, sort of the standard, just grab a probiotic, prebiotic, postbiotic, all that stuff may not be terribly effective in the end. Is that right?
It depends on how you think about it. There's a lot of clinical trials that have been done on, say, high fiber diets, on fermented foods, various probiotics. Many of them do show a significant effect on some phenotype, but inevitably there are responders and non-responders to all of these interventions. This isn't just true of the microbiome. This is also true for pharmaceutical drugs, right? Um, and so what these precision algorithms are helping us do is to rescue the non-responders. There's always a subpopulation that would benefit from a different intervention. And if you can a priori rationally predict what those are, you essentially rescue those non-responders. I mean, I would say, like, most people, general advice of eating a whole food, high plant-based diet that contains a lot of fiber, eating fermented foods. If you follow those rules, you're probably going to be doing pretty okay. But in our models, there are still some people that don't respond all that well, even to those interventions.
Sure. So there's a lot of buzz these days about basically fecal transplants, right? Um, from everything from gut disorders. Gosh, I've read people talking about autism, which I don't think that's probably something that has that much scientific rigor to it. But what's the real promise of fecal transplants? I mean, obviously, you talked about how your delivery of butyrate directly through enemas seems to be beneficial. The idea that here, I guess, is taking somebody who has a so-called sort of very healthy microbiota and transplanting, basically just transferring some of their poop over and seeing if you get a similar microbiota. Tell us a little bit about that.
Yeah. This is actually the first case of, clinical translation in the microbiome space. So I did my postdoc with a guy named Eric Alm at MIT, and he started the first stool bank in the country called Open Biome. At the time, they were using fecal transplants to treat a condition called recurrent Clostridioides difficile infection, or recurrent C. diff.
Yeah.
And the problem with that was that every hospital, every doctor performing those procedures had to go out and recruit a donor for their patient to give a fecal transplant. But most of us walking around in the world carry opportunistic pathogens in our guts. They're just suppressed by our endogenous microbiota. But if I was to give that to a sick person, that could make them even sicker. And so these hospitals would have to screen dozens of patients before they find the one magic person who kind of lacked any of those opportunists. So what Open Biome did is it turned it into a factory where they found these healthy, pristine people, and they just had them poop every day for a year, made huge batches of their material, turned it into pill form, and then shipped those out to hospitals, making it essentially very, very easy for these doctors to perform the procedure. The clinical trials on this are crazy, right? It's one of those where they have to stop the trial early for ethical reasons because the treatment is so much better than the standard of care that it would be unethical to continue giving people antibiotics. So it's something like, depending on what trial you looked at, between 85 and 99% efficacy for treating recurrent C. diff, which used to be kind of a death sentence back in the day. So super effective. And also now there are 2 FDA approved fecal transplant equivalent interventions that you can get from the doc that are healthcare reimbursable.
But is limited to recurrent C. diff infection. It seems to me that, again, we're focused on things that you can do to stay healthy. And are those things that are potentially available to somebody who's just, who's got a bad microbiome, who needs sort of a reboot, so to say? Is that stuff being studied in that way at all? Has it been looked at?
Yes, people have studied this in many different ways for many different conditions. There's a lot of trials for, inflammatory bowel disease or irritable bowel syndrome or autism or various things, right? And the outcomes of many of those trials have been very middling, right? There's been no example of such a stark effect size as what we saw for recurrent C. diff. So these FMTs seem to work pretty well for pathogen suppression. So if you have some opportunistic pathogen in the gut, these ecosystem transplants do really well. But for a lot of these other diseases, it seems like it's just a hammer, right? You're throwing at the system, and it's not necessarily doing what you want it to do. Something more precision is likely going to be required for many of these other conditions. And then, there's not a lack of risk for these procedures, right? So like I said before, there's been a few cases where patients have actually got sick from fecal transplant material. It's very rare, right? These are generally very safe interventions, but occasionally you can take up a pathogen. And then there are these other weird side effects that happen. Like some people show that depression is transmissible via fecal transplants or other kind of gut brain things. And like you mentioned autism before, that's actually one area where some of the recent data is looking slightly promising. There's actually a few open label trials that have shown some symptom amelioration in kids with autism. I think a lot more needs to be done to flush that out, but where these will be applicable and where they won't, I think there's still a lot of work and science being done around that. But generally speaking, it's sort of a hammer. And I think the main application we've sort of found, the lowest hanging fruit is picked. And now it's moving on to more mechanistic and targeted interventions.
Yeah, I mean, I think from a longevity standpoint too, I think you guys have, found that there are different types of chemicals. You've mentioned butyrate already, but you've also discovered gut microbes that may influence how well drugs like statins work, right? So what do you think we're sort of closest to becoming part of everyday medicine with some of these new techniques?
Well, it's a good question. It's actually surprising how many domains there are clinical trials happening in with the microbiome. One of the big ones I think that's very promising, and we'll see something in the next few years, is precision nutrition.
Mm-hmm.
There've already been a few companies that have come out. Day2 from Israel, and then there's Zoe in Europe. They both have precision nutrition algorithms that leverage microbiome data to predict blood glucose responses to diet. And that's based in solid science. Zoe, I think, still exists. Actually, Day2 went out of business, but you can now purchase their service, right? So people can actually track their blood glucose responses and diet from the microbiome and get predictions. There's a lot of snake oil out there, but that's one of the companies I think doing it better than most. But then there's a bunch of, like, NIH has this big push now for precision nutrition, where they're doing these huge clinical trials where they're collecting microbiome data, doing dietary interventions. And I think the outcomes of those trials will be used to build these models and algorithms to make these predictions. And I think our lab is contributing to this as well. So that's a big area. Another area that's something to look out for is cancer immunotherapy, right? So one thing that the microbiome is really good at is modulating your immune system.
Yeah.
Butyrate is a good example. It actually affects your T cell differentiation and kind of systemic immune state. And there's a lot of great evidence that fecal transplants or probiotics can actually influence the outcomes of immunotherapy and augment, kind of take non-responders and convert them to responders. That's super promising. The first defined cocktail of microbes for treating recurrent C. diff is now in phase 3 trials by this company called Vedanta. So this is supposed to replace fecal transplants for recurrent C. diff treatment. I think, if that phase 3 trial goes well in the next year or two, we should see that as an FDA approved therapy. But there's a bunch of stuff.
Is that with microbes? So with basically like a soup of probiotics specifically?
8 strains of Clostridia. So it's a probiotic.
Got it. Interesting stuff. Well, I guess one last question for you. I guess people listening out there wondering in general what your take on, obviously we talked about high fiber diets and that kind of thing, but do you have an opinion on, people should be taking probiotics, postbiotics, prebiotics, anything like that, just as a supplement or not so much?
Yeah. It's a good question. I mean, they usually can't hurt, right? One of the issues with a lot of these probiotics is that, you look at all these clinical trials, there's some efficacy shown, and it's a very specific strain. So you have to maybe go track down that particular strain of Bifidobacterium infantis if you want to kind of have an apples to apples for what you're taking versus what that clinical trial showed. So there's a little bit of murkiness in mapping what's out there in the science to the products that you can actually grab off the shelf.
Yeah.
So that's something to keep in mind. Another thing to keep in mind is most of the probiotics that we have available to us are grandfathered in by the FDA. They are what are considered generally regarded as safe or GRAS because they are already present in food, right? They're the same microbes that you find in yogurt and in sauerkraut and in kimchi. So they're considered food, and there have been no clinical trials to prove they're safe. They just let you eat them because they're already in food. But the dominant microbes that actually live in the adult human gut microbiome that I think would be useful for actually treating disease and helping people, those organisms, I can't just take them out of me and give them to you as a pill.
Yeah.
Like Fecalibacterium presnitzii, for example, is a butyrate producing Clostridium that was very dominant in the human gut, but some people lack it. And if only I could give them that organism, I think that would do them a lot of good. But you have to first run a phase 1 clinical trial so that regulators won't slap your hand, because you have to prove it's safe before you give it to someone. And that costs $1 million to run those trials. And so I think getting these next gen probiotics into the market will require significant investment from companies, which may lead to a slight rise in the price of these products on the market. But I think that's a price we'll have to pay to have more effective probiotics. There are a few of these that already exist on the market. There's a company, Pendulum, that sells a few of them, like Akkermansia and some of the Clostridia I was talking about. And I think there's a couple other companies in Europe that sell similar products. But these kind of organisms like Akkermansia and butyrate producers, if they seem to be on the decline in older people who are less healthy. So if I was going to tell anyone to take probiotics, I would say people who are over 50 or 60, maybe start taking these cocktails of like Akkermansia and Clostridia as a preventative measure to try to maintain, metabolic health as you age.
Yeah. Interesting.
But also eat a lot of fiber and get good exercise. And that's probably the biggest thing you can do.
Correct. Sean, where can people learn more about this stuff if they're interested and they want to maybe find out, some of what's going on, see if it applies to their own situation, that kind of thing?
Yeah, you can follow our research at Gibbons, Gibbons .isbscience.org. That's the website.
Fantastic. Thanks so much for being on the show today.
Thanks for having me.
Thanks for listening. A quick reminder that while I am, in fact, a surgeon, nothing I say should be construed as medical advice. Now, make sure to include your physician in any medical decisions you make. And also, if you're enjoying the show, please make sure to show your support with a like, share, or subscribe.